afternoon, all. Myself, Swapnil Chavan. I am a computational toxicologist at RISE. I lead innovative research in the field of predictive toxicology by using AI, chemoinformatics, bioinformatics, and image informatics techniques. It's my honor to present my team's work in today's webinar. My subject of area will be automation of a predictive toxicology. We all live today in a digital world where we enjoy smartwatches, smart TVs, smartphones. We have a home automation and a self-driving car is on its way. This digitalization and automation has also impacted our pharma sector. And my today's talk will be centered around automating the field of predictive toxicology. I will be talking about traditional predictive toxicology. What are the obstacles in this traditional way of doing predictive toxicology, and how AI will help us in overcoming those obstacles? So, without a further ado, I will move on to my presentation. So, I will be talking today about our AI-TOX suite. It's an end-to-end solution for automated predictive toxicology with the help of AI. So the main founding stone of a predictive toxicology was placed in the year 1961 to 69, where the field of physical organic chemistry saw a big revolution with the invention of a QSAR. During 1961 and 62, Hanch and Fujita and their team, they come up with uh, correlating the biological activity with the help of few electronic constant and few partition coefficients. Later on in 1969, Hanch has expressed this correlation of biological activity in the form of a mathematical expression, and this was known as the first QSR model. Uh, with the advancement in computer hardware and software, this small mathematical expression is now transformed to a more complex form with more computational power in hand. So this is how in a current uh, world this traditional QSAR modeling is done. uh, the chemical structures, either in the form of mole file or SDF file or a smiles notation, are being used, and then they feed to a software where these smiles or structures are converted to some numerical numbers, and these numbers are called as descriptors. Afterwards, these descriptors are then further refined. If they are highly correlated, few of them are dropped. They need to be scaled, they need to be centered, and these all experiments are known as feature engineering. After feature engineering, these descriptors are used to fit the model. For example, equation of line. And then this model is used for a forward prediction, either a biological activity or a toxicological activity. What happens is uh, when this main molecule is being transformed to this numerical descriptor, this transformation is not foolproof. There are flaws in this process, as well as when these numerical descriptors are undergo feature engineering, there are few issues. And I will be walk you through with what are the problems this traditional way of QSR building face. The first shortcoming of this descriptor is they can't be derived for a mixture and salts. So let's take an example of a lead acetate. When one need to use a lead acetate for a QSR modeling, they need to convert this structure to a QSR ready structure. And this QSR ready structure can't handle a ionic bond. So your lead acetate is converted to a acetate. And while doing this QSAR ready transformation, you this is a mutagenic structure. You end up converting this mutagenic structure to a non-mutagenic structure. Second drawbacks with this descriptor is the when your molecules have a carbon, hydrogen, oxygen, nitrogen, it's much easier to calculate these descriptors. But if your structure has non-common elements like lead, antimony, barium, beryllium, mercury, chromium, the softwares cannot able to handle this, and a lot of these descriptors are having a missing values. So that's the second disadvantage. They can't be derived for a non-common elements. The third disadvantage is while handling the large structure, such as microlite antibiotics. Such a large structure, their descriptor calculation could be difficult and a time consuming. The fourth drawback with this descriptor based QSR is one need to do a pre-processing of the descriptor and a feature engineering like scaling, centering, then removing the highly correlated descriptors, removing the zero variance descriptor. So this feature engineering is an essential step in the traditional QSAR, but that needs a time and a manual steps. The fifth disadvantage is the number of available descriptor are limited. For example, for a numerical descriptor, the AlvaDesk software, formerly known as Dragon, has approximately 6,000 maximum descriptors. And for a binary types of descriptor, there is a Pharma Gobi 2D descriptor which has close to 40,000 bit string. And this is a limit because in a traditional QSR, there is a rule of 1:5. So for a 5 chemicals, you need a one descriptor. That means if you have a maximum 6,000 descriptor, you could use this to model up to a data set of a size 30,000 compounds. But if your data set is higher than that, then you don't have enough descriptor to model such a big data, and that's the fifth disadvantage. The sixth disadvantage is sometimes they can be identical for a dissimilar molecule. What I mean by that is here are the polybrominated compounds, and if you calculate a pubchem fingerprints for these four compounds, they all those will be exactly same. So imagine if someone is using PubChem fingerprints for these polybrominated compounds, they are representing same compound for a very dissimilar molecules. And that's a big drawback. One could argue, okay, PubChem fingerprints are a binary and it could be possible that they can have identical pattern for a dissimilar compound. What about numerical descriptor? Then answer is yes or no. So if one could talk about a single numerical descriptor, let's say a melting point, you can find definitely chemicals where two chemicals have a same melting point. Two chemicals can have a same log P values. So they can be same, but if you look for a set of property numerical descriptors, then they might not be identical. What about in case of tautomers and stereoisomers? Well, in that case, they could be likely exact identical. One need to figure that out. So the identical descriptor is the sixth major drawback with the descriptor-based methods. The seventh drawback is they are non-reversible. What I mean by that is if you have a structure and if you run this through some descriptor calculation software, you will end up having a table of a descriptor, let's say molecular weight, log P, hydrogen bond, acceptor, donor, TPSA. But if someone give you this table of a molecular weight, log P, hydrogen bond, acceptor, donor, TPSA, and ask you what is the original structure, one could not trace it back to the original structure. So they are a one way, they are non-reversible in nature, which is a kind of drawback. Uh, the lastly, the mechanistic interpretation. So once someone use this kind of a descriptor for model building, But if one need to interpret the outcome of a model, then they need a domain expert who could able to understand what is the log P, what is the hydrogen bond acceptor, and then he could use his domain knowledge. For example, here, if there are two oxygen, and that's why they have a hydrogen bond acceptor as a two, and these two are forming some kind of a uh, hydrogen bond accepting activity within a structure, that therefore it's been seen as active or inactive. So without such a domain knowledge, one could not do a interpretation. So requiring a domain knowledge is another big drawback with these traditional QSR methods. So one could ask, can we just not do without a descriptor? In another way, is there a way where one could do a descriptor free modeling? And today I'm gonna present you what research we did within the RISE, where we have used two different descriptor-free approaches to build our in-house models. So representation learning is last decade's biggest revolution we saw within the pharma field, where the graph-based technique and a text-based techniques are very popular. The graph embedding based deep neural networks are the models. They transform the molecules to some kind of a graph where every atom is considered as a node and every bond is considered as an edge. And once this molecular structure is transformed to a graph, then they convert it to some kind of a one hot encoding, which means if it's a yes and no table, if there is a nitrogen present at a position 1, if it's yes, it's given a number 1. If it's no, then it's given a number 0. So that's where we get the one hot encoding as 1 and zeros. Same applies to the edge embedding. If the bond is present or absent, what is the hybridization of bond? So in that way, they given a one hot encoding. And then this one hot encoding is attached to a sequence of neural network layers. And after a model training, one could able to predict whether the given compound is a toxic or not. Uh, Second approach is a text embedding based deep neural network. So in our pharma field, we use a smiles notation as a text. So what it does is it uses a standard library of all periodic element uh, as a table, a lookout table. And then for every character within a smile, we look for this lookout table and assign the given index of the given character. So for example, the C is at the position 1. We, we transform the first C as a 1. Again, second C, 1. The oxygen will be given 2. The double bond will be given 5. So in this way, we transform this text to a numerical number, and that's known as a text embedding. And then this text embedding is fed to a sequence of neural network layer. Then we train the model, and that way we come up with the probability whether given compound is a toxic or a non-toxic. So these are the two methods that we have, which is known as a descriptor-free methods. And what today I'll be talk about this second method, the text embedding based TNN. So now the first advantage we got is we got rid of descriptor. The second advantage is it provide a ease of quantifying the uncertainty. In a neural network, it's very easy to quantify uncertainty. The neural networks are a sequence of layer which consist of nodes, and a lot of mathematical transformation happens within the node which are fed to the next layer until the last layer. What uncertainty quantification looks is it null the certain nodes input and output. By doing that, it stop model to look the same pattern time and again. By nulling the different operation at the various iteration while model training, it does not depend on seeing the same pattern. This is what we call a regularization. And by doing this regularization, while in a in a forward prediction step, one could able to derive what is the uncertainty within the model. So that's the second advantage with this descriptor-free approach is they are easy to use for quantifying the uncertainty. The third advantage of these descriptor-free methods are so one could use the text to transform them to a text embeddings, and these embeddings then attach to the neural network to predict the toxicity. This process is a reversible. That means one could use certain explainable AI techniques and use the predicted outcome and trace it back through each layer of a neural network all the way up to the input and see which part of input has a highest contribution for the given predicted output. And the explainable AI is a newly developed field which help one to understand what part of the input was valuable and contributing towards the given prediction. It also allow one to debug the model if model is looking at the wrong structure or wrong part of the structure. So this was the third advantage of a descriptor free method. So lastly, I'm going to show you one of the example from our AI-TOX model suite. This example is the mutagenicity model. This model has been trained with quite a large mutagenicity data set come from a Helma et al.'s 2021 paper. We use recurrent neural network, the text embedding best approach to construct the model. And for model explanation, we use integrated gradient method. The model results looks like something this. For a twice repeated tenfold cross validation, model has shown AUC value of 87%. The balance accuracy is 81. The sensitivity and specificity all are above 80%. For a external test set, also models showed quite robust output with the AUC of 87%, the balance accuracy 79, accuracy 79, and sensitivity specificity close to 80%. So was a quite robust model. And then we deploy this model to a web server. And this is how the terminal of a web server looks like. So it just need input in the form of smiles. So let's say this is a cyclophosphamide, and in the form of smile notation, it looks something like this. So one need to just feed in this query to the given section of a terminal and then press run. Once that's done, this is how the model gonna spit out the output. So the output will be consist of the prediction, whether it's mutagenic or not. Then the second information it will provide is the uncertainty, how confident the model is for labeling given input as mutagenic. So here model has shown 100% confidence. And the third advantage is it comes with explanation. So it shows a toxicophore. So for the given cyclophosphamide, it's an alkylating agent where a phosphoric mustard play a big role. And our explanation has highlighted that these functional groups were very important while labeling this compound as mutagenic. Another important substructures was this nitrogen mustard, the nitrogen with the two halides. And as all of you know, the nitrogen mustard is an important alkylating group, and that could cause a mutagenicity. So this was a very good example how the model not only do the prediction, but it also provide the uncertainty and the explanation why they predicted something mutagenic. So one more advantage of our text-based embedding is we could not only limit to the structure, but it can also be applied to a reaction. So here is a reaction of a cyclophosphamide synthesis. This is a cast number one could look for in a SciFinder. So it has this oxazophosphorin derivative along with the dichloroethane, which lead to a formation of cyclophosphamide. What I did in a input is I used this first group, and then with the full stop, we added the dichloroethane as a second group. And Using this input, it's a SMART format of a reaction. Uh, our model has predicted this as a mutagenic with slightly low confidence, but with a true output, that is the mutagenicity. So these AI models are not only limited to a single compound, but they could be extended to a reaction, which a traditional method cannot do. Traditional QSAR can't handle such a ionic bond. They can't handle a reaction. They can't handle a mixture. And the model is smart enough that it knows these are the reagent for a given reaction, and they have a component that could cause a mutagenicity. So model was quite smart. And with this reaction, what usefulness of this new text embedding methods will be, they could be widely used in a life cycle assessment field where there is not one component related to the compound, but there are his pre-formulation components and their degraded product of the given compound. So this text embedding methods has a huge potential playing a role for a life cycle assessment. With that, I summarize our AI-TOX tool. So this suite has consist of human toxicity endpoints like CMR, carcinogenicity, mutagenicity, reproductive toxicity, as well as heart, liver, eye irritation problems. Then there are 12 endocrine receptor-based models. There are 3 ecotoxicity related models. So in summary, uh, a QSAR model should be predictive, should be certain, and should be explainable. With our AI-TOX models, they fulfill all of these 3 criteria, which make them a good models. The second advantage what our models have is they can be applied to salts and mixtures, which is not possible with the traditional QSR methods. And the lastly, our models could be applied to the reactions also, and we have screened those for human and environmental hazards. So we see a huge potential of this our text embedding based AI models towards the life cycle assessment. So lastly, I would like to acknowledge my colleagues, the Eric Ilippa, who is my former colleague from the Intelligence System Unit at RISE, then Maria, currently a PhD student within the RISE in an Intelligence System Unit, Professor Dr. Ian Cotgrave, our senior researcher at a Chemical and Pharmaceutical Safety Unit in RISE, and Swarke Janssen, the steering committee member of a AI-TOX project from a computer system unit in a RISE. I would also like to thank RISE for funding this project via SK Medal funding and the Mistra SafeChem project. Lastly, I thank you for your attention, and I'll be happy to answer your question either now via face-to-face, or you could also email me via this email ID. So thank you. I close my presentation now.